Hello, this is Chapter 27 review. It's all about medically or clinically significant fungi. So, this fungi cloud includes yeasts and molds. I don't want you to be confused if you see two different spellings of the word mold in your PowerPoints. M-O-L-D is how you probably know it to be. And this is the American spelling for all senses of the word meaning. However, you'll see it as M-O-U-L-D in your PowerPoints, and this is the British spelling. So both spellings are the same, mean the same. So anyway, let's just generally speak about fungi. Some general characteristics. They are eukaryotic cells. They possess a true nucleus, nuclear membrane, and mitochondria. Their cell walls are made of chitin. Most are aerobic, growing best at a neutral pH, and moisture is a necessity for growth. Although spores, or also known as or also called conidia, can survive dry conditions. So when we compare yeasts and molds, yeast are single vegetative cells that produce pasty, creamy colonies on agar. They reproduce by a budding, and they produce these daughter cells called blastoconidia. Molds, on the other hand, are fuzzy, woolly colony appearance on agar, and they produce something called mycelium, which is made up of hyphae. Hyphae can be aerial, which means it grows up above the agar surface, or hyphae can be vegetative. Vegetative meaning that it grows down into the agar, kind of like roots. The shape of the hyphae can aid in identification for a lot of these species, so it's important when looking at the big picture clinically, if you have a mold present, what the hyphae look like. Also, molds contain root-like structures called rhizoids. These help the mold absorb nutrients from whatever it's growing on. We can further kind of break molds into two categories. And one is based on the type of hyphae they produce, and one category is based on their color. So if we look at how their hyphae grow, they can be classified in three ways. They can be called septate, they can be called sparsely septate, or they can be aseptate. So mold hyphae that are septate have cross walls. Their hyphae have cross walls or septa in between the cells. Aseptate is a hyphae that have no cross walls. And then sparsely septate hyphae are cell walls that are far and few between. So if we look at color, the color grouping, we have something called hyaline or dematiaceous. So a hyaline mold has lightly pigmented hyphae. In a wet prep, these would appear clear. Or we have dematiaceous hyphae, and these are darkly pigmented hyphae due to the presence of melanin in their cell walls. So these would appear brown or black, or possibly, if stained with lactophenol blue or lactophenol cotton blue, they may appear a blue color because that lactophenol cotton blue stains the chitin in their cell walls. So that's kind of a few terms there that you need to be familiar with. So let's talk about another very important term called dimorphism. So some fungi have the ability to exist in two forms. It just depends on the temperature that they're growing in. So if a fungus is able to grow at 37 degrees or in vivo, which most most pathogens are able to do. A dimorphic fungus is called a yeast, or has a yeast or spherules phase when growing at 37 degrees. Now, a dimorphic fungus has a mold phase if it's seen or grown or able to grow in room temperature, which is 25 to 30 degrees. So dimorphic fungi are able to do both. They're able to exist in both forms, a yeast and a mold phase. So just know that most pathogens are dimorphic fungi. Fungi reproduction includes two different forms as well. There is an asexual form of reproduction and a sexual form of reproduction. A fungus that reproduces asexually is termed anamorph, and it results in the formation of conidia. Conidia is another or synonym term for spores. So an anamorph that asexually reproduces forms conidia spores following mitosis. Conidiogenous cells can produce two different shapes, primary shapes of conidia. And I just want to mention those because these are a couple more terms that you'll need to know. The first shape is a phelid, and that's kind of like a vase shape. And then the second shape is an annelid, and that's like a ringed structure. So conidiogenous cells produce two different forms of conidia. There's also something called arthroconidia, which is a form of conidia that's formed by fragmentation of fertile hyphae. So a segment of the hyphae kind of breaks off, and that is considered an arthroconidia spore. So then let's move on to sexual reproduction by fungus. And so a fungus that reproduces sexually is termed telomorph. This requires the joining of two compatible nuclei followed by meiosis. And the result is the production of a zygospore. So like a conidia, these are zygospores. So know the differences in sexual and asexual reproduction. Okay, so let's move on. This chapter, by the way, is very detailed. It has a lot of information. You can take a college course that is just covering this information, mycology, the study of fungus. So know that this is a huge, huge amount of information that we're just kind of cramming into one little chapter review so that you have some familiarity with it and what you might see in a clinical laboratory. So there are a lot of details in here. So just take your time when studying these and maybe create a flow diagram to keep all of the differences and, separated. Try and keep, find some way to organize all this information because it is a lot of information. Okay, with that said, there are four major divisions of fungi. There are zygomycota, ascomycota, basidiomycota, and then something called fungi imperfecti or imperfecti, and this is the deuteromycota. The deuteromycota is the most commonly seen group. This is what you'll see in the clinical lab the most often. A little bit about each of these different major divisions. Zygomycota also causes zygomycetes. That's the infection type. These are rapid growers found in the soil. They have an asexual reproduction that's characterized by specialized hyphae called sporangiophores. Sporangiophores produce spores that are termed sporangiospores. Sexual reproduction occurs with the production of zygospores for this particular group. Some examples are Mucor, Rhizopus, and Absidia. So these are a few clinically significant Zygomycota species or genre. The next major division, which was Ascomycota, this one has sexual reproduction that's characterized by the production of ascospores. So ascospores are produced in a sac-like structure called an ascus. Usually, though, we identify Ascomycota species based on asexual structures. Some examples in this group or division of fungi is Microsporum and Trichophyton genre. The next major division is the Basidiomycota. There are just a few clinically significant infection causers in this division. There is one true pathogen, and it is Phylobasidiella neoformans. And this is the sexual form of Cryptococcus neoformans, variety neoformans. So know that Phylobasidiella neoformans is the sexual form of Cryptococcus neoformans. So it is just given another name in its sexual form. It's a big clinical concern, Cryptococcus neoformans. So know that. It's difficult to ID this particular specimen in the lab because these are non-sporulating organisms, or sterile is what they call them when isolated in the labs. The only thing you really see are hyphae or hyphal elements, so it's kind of difficult to diagnose those. Let's see. Deuteromycota, that last fungal division, the most commonly seen, also called fungi imperfecti, or imperfecti. This includes the largest group of fungal organisms. The range of infections is from superficial to deep tissues to even systemic infections or systemic mycoses. There's no mode of sexual reproduction identified in these, and ID is based primarily on asexual reproductive structures. So that's a little bit about each of the major 4 divisions. So we're going to move on to fungal infection types or mycoses. There are 4 groups of fungal mycoses. And it's based on location in the body. The first fungal mycosis type is superficial. The second is cutaneous, or we call this dermatophytosis. The third big group is subcutaneous infections, and then the 4th big group includes systemic infections. This can also include deep tissues, lungs, and other organs. So we're going to talk about each one of those mycoses types. So let's start with the first one. The first one is superficial mycoses. These occur in the outermost layer of your skin, cornified areas. This would be the stratum corneum of your epidermis, and most of the time it's just cosmetic effects. There are 4 primary species that we'll talk about. The first one is Melissacia furfur. This causes something called tinea versicolor. We know it as dandruff. Its hyphal elements and conidia or spores have the appearance of spaghetti and meatballs. Unique little trait. The second superficial mycosis agent is Piedra horte, and this causes something called black piedra, and this affects your scalp and your hair. You'll have little black gritty nodules on your hair shaft with this particular mycosis. The third species is Trichosporin beigeli, and this causes something called white piedra, and this affects your face, facial, and genital hair. And with this one, you have white mycelial mat, a white mycelial mat around your hair. And then the fourth species of superficial mycosis is Phaloanellomyces werneckii. This causes something called tinea nigra, and this primarily is displayed on your the palms of your hands and the soles of your feet. Okay, so let's move on to the second type of mycosis, and this is cutaneous mycoses, also known as your dermatophytosis. These are keratinophilic, so they love hair, skin, and nails. Keratinophilic. There are three genera that I'm going to talk about. The first one is the Microsporum, the second one is the Trichophyton, and the third one is the Epidermophyton. I want to say something before I continue. The term tinea just refers to disease state. So when I mention the next 5 dermatophytoses, they all start with the term tinea. So it just means the disease state. So your 5 primary dermatophytoses in this category of cutaneous mycosis are something called tinea capitis, which affects your head, tinea barbae, which affects a beard, tinea corporis, which affects the body, tinea cruris is affecting the groin, and this is also known as jock itch. Tinea pedis is affecting your feet, and this is also known as athlete's foot or moccasin foot. Moccasin foot it was termed this because usually you'll develop the mycosis in the webs in between your toes and the web of your foot. So it was given the name moccasin foot. So your 3 genera. The first one is Microsporum. I'm just going to mention 3 organisms that are in this that can be clinically significant. Microsporum canis, Microsporum gypsum, Microsporum auduinii. So just be familiar with those. The second genre is Trichophyton, and there are 3 specifics I'm going to talk about in this group. But real quick, I'm going to move on to the third genre, and that's Epidermophyton. And I just want you to know of one particular that you may come across in the future, and that's Epidermophyton floccosum. So let's go back to Trichophyton. This is the only genre we're really going to discuss a little bit more detail. There's 3 species I want to talk about. The first one is Trichophyton mentagrophytes. This is the most commonly isolated dermatophyte. It infects skin, hair, and nails. It produces conidia, but I haven't mentioned this yet, but there's 2 terms, macroconidia and microconidia, I want you to be familiar with. Macroconidia are spores or conidia that are more detailed and complex, larger. They're larger spores. And then you have microconidia, which are usually simple, single-celled. So when I mention those 2, you'll know what I'm talking about. So with Trichophyton mentagrophytes, it has a macroconidia, or produces a macroconidia that's cigar-shaped. That's about 4 to 5 cells long. And then it also produces microconidia that are globose, or just round, single-celled. The next species, Trichophyton rubrum, this primarily affects your skin only. The colonies appear burgundy or wine colored. They produce both macroconidia and microconidia. The macroconidia are rare, but the microconidia have this clavate, tear-shaped, or upside-down wine glass appearance. So know that. And then the third species is Trichophyton tonsurans. And this can affect skin, hair, and nails. It's the leading cause of tinea capitis in children. So that is the disease state of the head, capitis. And we also know this as cradle cap on babies. So this is the cradle cap causer, Trichophyton tonsurans. They do not produce macroconidia. They only produce microconidia that are peg-shaped. And a unique fact about this one is that it has an absolute requirement for thiamine. So one of the tests you can use in the laboratory for identification of fungi, yeasts, and molds is thiamine requirement. So this is one way that you could test for this particular organism. So that's your 3 genera of cutaneous mycoses or dermatophytosis. Okay. Oh, one more important note. I think I accidentally skipped this. So cutaneous mycoses that affect your hair are can be also described as an endothrix or an ectothrix fungal growth. An endothrix is a fungal growth inside the hair shaft. An ectothrix fungal growth is one that's on the outside of the hair shaft. And then when referring to fungal infections of the nails and hair, nail beds, these can be called onychomycosis. So there's some other alternate terminology terms that can also be used. I just want you to be familiar with those. So let's move on to our third big group or type of mycosis, and this is the subcutaneous mycoses. These primarily affect subcutaneous tissues, so deeper tissues. Usually they're the result of a traumatic implantation into deeper tissues of the skin. These are found in soil or organic decaying material. There are 4 primary diseases in this large subcutaneous mycosis group. The diseases are chromoblastomycosis, eumycotomycotomas, phaelohyphomycosis, and sporotrichosis. Sorry. They'll definitely tongue tie you. So let's talk about the first disease type, and this is chromoblastomycosis. These are chronic mycoses. A chronic mycosis can last months or years. With this particular disease type, you develop lesions on your extremities. They're known as cauliflower lesions. One particular organism that causes chromoblastomycosis is Philophora verrucosa. This particular species has a vase-shaped phyllid with oval conidia at the ends. The next disease type is a eumycotic mycetoma. These cause indolent infections. So indolent is just meaning that it has little or no pain related to it. So in mycetomas or chronic infections, these affect your skin. They are pus producing and often have a sinus tract or create sinus tracts for pus draining. You have an organism, a species called Pseudo-Alisteria boydii, and this is a sexual form. It also has an asexual form, but the asexual form is given another name, and its second name is Pseudosporium apiospermum. So just know with this particular organism, Pseudoalasteria boydii, that it has a second name in its asexual form. The third disease type, or subcutaneous mycosis type, is the phaeohyphomycosis, and these are dematiaceous organisms. So they have dark colored hyphae, and these are found ubiquitously in nature. One particular species is Wengiella dermatitidis. These produce black colored colonies. And then the fourth disease type in the subcutaneous mycosis group is sporotrichosis. Sporotrichosis is associated with gardening. One particular organism called Sporothrix schenkii causes something called rose handler's disease. It's a dimorphic fungus. At 37 degrees, it produces cigar-shaped conidia, and then at 25 degrees, it produces rosette-shaped conidia. So very interesting. Okay, so that brings us to the fourth group or type of mycoses, and that's systemic mycosis. Excuse me. So all of the systemic mycoses are dimorphic fungi or caused by dimorphic fungi. Systemic mycoses are often fatal and very serious. Most of the agents that cause this type of mycosis are ubiquitous in the soil. Usually they're acquired by inhalation. So infection is caused by inhalation. And usually the primary infection site is your lungs or respiratory tract. There are 4 classic agents causing systemic mycoses, and then one that's kind of an opportunist, but that it's also important. And then there are several others that are opportunistic that I'll mention briefly. But the 4 main or classic agents I want to talk about are Blastomyces dermatitidis, Coccidioides immitis, Histoplasma capsulatum, Variety capsulatum, and Paracoccidioides brasiliensis. So that first one, Blastomyces dermatitidis, this causes blastomycosis or Gilchrist's disease. It is endemic in the Mississippi and Ohio River basins. So where all these are dimorphic, they both have a mold phase and a yeast phase. So I'm going to mention about both of those for each of these. The mold phase with Blastomyces dermatitidis has white tan colonies, short hyphae with single-celled conidia. They look like lollipops. The yeast phase of Blastomyces dermatitidis is broad-based, thick-walled, double-celled. You can see these in sputum gram stains. So they're broad-based. They kind of have like a circle next to circle appearance in the gram stain, but they're very broad. The next specific organism causing systemic mycosis is Coccidioides immitis, and this particular organism causes coccidioidomycosis, or valley fever, or desert fever, or Sanoquin fever is another name for it. This one is probably the most infectious fungal agent just by breathing it in. Its mold phase has aerial mycelium. Its vegetative hyphae produce thick-walled, barrel-shaped arthroconidia that alternate with empty cells. Its yeast phase, or you can see this yeast phase in tissues, human tissues, are large, round, thick-walled spherules. Okay, the next organism is Histoplasma capsulatum, variety capsulatum. It's kind of like a biovariant. So this organism causes histoplasmosis, or also known as Cave's disease or Darling's disease. Primary site of infection is in the respiratory tract. This one, however, is endemic south of the Ohio River and east of the Mississippi River. This organism resides in the soil with high nitrogen content, so it is especially likely to find this organism in soil that contains a lot of bird droppings or bat guano, giving it the Cave's disease name. So the fourth organism that causes systemic mycosis is Paracoccidioides brasiliensis. And this causes paracoccidioidomycosis. That's a mouthful. Paracoccidioidomycosis. This is also called South American blastomycosis or Brazilian blastomycosis. Its yeast phase has large cells with multiple budding, kind of looks like a mariner's wheel or a Mickey Mouse cap. Its mold phase is septate, has branched hyphae with terminal chlamydospores. Okay, so that's your 4 classic agents. There is another agent that's worth mentioning that's up there on the importance list, and that's Penicillium marnefi. This is a dimorphic fungus as well. Its mold phase, you'll see white to green colonies with soluble red pigment that diffuses into the media. And then the yeast phase of this particular organism has sausage-shaped cells. So just know those characteristics. I'm going to mention a few opportunistic fungal pathogens and know a few characteristics about each of these. The first one is the genus Aspergillus, and this is the most commonly isolated agent in the clinical lab. Genre, most commonly isolated genre. These have swollen vesicles with swollen vesicle felids with chains of phyloconidia. They appear like a dandelion, so they look like a dandelion. Another genre, Penicillium. These are tightly arranged felids with conidial chains, so these appear like a bony fingers. They really kind of look like fingers. Then we have a genre called Alternaria, and this is a dermatiaceous genre, so it's pigmented, and they produce racket-shaped macroconidia. So they look like a tennis racket or racquetball racket. We have a genre called Rhizopus. These are dark, they have dark sporangia with rhizoids joined by something called a stolon. Then we have a genre called Fusarium. Fusarium are a hyaline fungus, so they're very, very faintly pigmented. They have a sickled shape or a canoe-shaped macroconidia that are 4 to 5 cells long. Then we have another genre called Mucor, and this is similar to Rhizopus species, except there are no rhizoids with this one. So those are some opportunistic funguses. Okay. So one thing I want to back up 1 second that I didn't mention, I do want to mention to you all now. With your cutaneous dermatophytes, that second group of mycoses, the keratinophilic organisms. These also can be classified based on where they're found in nature. There are 3 terms related to this, and this is geophilic, which inhabit the soil, zoophilic, which are particularly infect animals, and then there are anthrophilic organisms or dermatophytes, and these infect humans only. So if you hear those words in relation to cutaneous dermatophytes, just be familiar, that's what those mean. Okay, so let's move on. We're almost done. I think we're going to briefly discuss yeasts. So yeast are primarily only seen at 37-degree temperature, right? They mostly reproduce asexually by budding. They're eukaryotic and they produce a pasty type colony. There are a few couple yeasts that I want to discuss. The first one is Candida albicans. This is the most common cause of yeast infections in humans. It's also the 4th most common cause of blood-borne infections in the U.S. There's a term called thrush, and when you hear this, it is an oral infection with Candida albicans. So thrush is the particular name or infection related to oral. There is a particular test that we use to identify Candida albicans, and this is the germ tube test. So a germ tube is the beginning of a true hyphae. Candida albicans are germ tube positive. They produce true hyphae. So a germ tube does not have a constriction at their base. So usually when you're looking under the microscope at a gram stain, you'll see a classic budding yeast, and that is not a germ tube. It is a budding yeast. It has a circle with another circle on top of it as it buds. Kind of reminds me of a bowling pin. But a germ tube does not have a constriction. It grows and grows and opens and grows further and further. So how we perform the germ tube test is we suspend yeast in serum and incubate it at 35 degrees for about 2.5 to 3 hours, and then we will observe under the microscope for germ tubes. If there are germ tubes present without any constriction, we can ID this as Candida albicans. It's the only one that is germ tube positive, or is an indication that it's Candida albicans. There's another yeast I want to talk about, and that's Cryptococcus genre. Cryptococcus neoformans is one in particular species that is an important cause of meningitis and pneumonia or pulmonary disease. So this organism, Cryptococcus neoformans, is commonly found in the soil that contains pigeon droppings, interestingly enough. The yeast of this organism, yeast phase, produces a capsule that can be identified by an India ink stain in CSF. And when we grow this organism on something called birdseed agar, it will produce a black colony. So a couple ways to ID this organism. Let's see. There are some other significant yeast species I just want to mention. Candida glabrata. This is the 2nd most commonly isolated Candida. Candida tropicalis, and then there's one called Rhodotorula, which is a species group. So Rhodotorula genre. They produce these bright salmon pink colony colors, so just a note worth mentioning. There is a Candida called Candida auris, and this is another rising concern in the clinical laboratory. It's very resistant to antifungals, and it's becoming a concern. So another one to know about, Candida auris. And then there's one more organism called Pneumocystis jirovecii, and this is an opportunistic pathogen, usually in immunocompromised patients or patients with AIDS. There are 3 stages to the life cycle: a trophozoite, a precyst, and a cyst. The cyst stage is the infectious stage. We would perform a lung biopsy, and if you find a cyst or trophozoite in that biopsy, it's diagnostic. Also, with this particular organism, a GMS stain, which I haven't talked about yet, but I'm going to here in just a minute. GMS is a methenamine silver stain. So methenamine silver, you will be able to see the cyst wall that stains black, and with the stain, the cyst will appear like punched out, or like a ping pong ball. So know that this particular organism has that appearance, a ping pong ball appearance. Okay, so we're going to wrap it up with just a few more bits of information about yeast and fungi and molds. A little bit about specimen collection and transport, some microscopy, some testing and susceptibility, and some medias. So to collect and transport specimens for testing that are when you're looking for fungus or yeast, we can collect body fluid. We can test body fluid. We can test CSF, especially with India ink stain, blood and bone marrow, sputums, urogenital and fecal specimens. Those first have to be centrifuged, and then the sediment is used to culture. Hair, we can directly plate hair on agar plates. We can use something called a Woods lamp, which is kind of an old school method, but a Woods lamp is used to detect fluorescent fungi. Oftentimes this Woods lamp is used in veterinary offices as well when looking for particular types of dermatophytoses. Skin, usually we use scrapings from lesions. We can look at skin under a wet mount. Most of the time when you have skin as your specimen source, we use 10 to 20% of something called KOH, or potassium hydroxide, and this dissolves the keratin in cellular material for better visualization of the fungal elements. And we can also use nails. We can look for fungus in nails. Usually we use scrapings and cuttings. Under the microscope, we can do a wet prep. We can stain specimens or smears with calcofluor white stain, which is a fluorescent stain, and it will bind to chitin, and you will have an apple green or blue-white color for positive fungal elements. India ink, remember that's the negative stain for CSF, looking for Cryptococcus neoformans. We also have a few tissue stains. We have something called PAS, or periodic acid shift stain. We have the GMS, which I mentioned, which is methenamine silver. We have Giemsa stain and Fontana Mason stain. So most funguses are treated with fungus or with an antifungal. This could include amphotericin B or some of the azoles group of antifungals, and this includes fluconazole. Susceptibility after isolation is not usually done or performed on yeast and fungus because there's a lack of established breakpoints for most fungal agents. However, there's a Vitek card out there for fluconazole only susceptibility testing. And then a brief mention of some medias we use to grow and culture fungal agents. The most common is a SDA, or sabro-dextrose agar. We also name this SOB. This is the SOB plate. There's a mycoseal, which is a SOB plate or SDA plate with antibiotics. And then there's also brain, heart, and fusion agar, or BHI agar, which is used commonly to grow fungus. We incubate our cultures at 25 to 30 degrees, most commonly for 4 to 6 weeks. And if there's a dimorphic fungus suspected, we also grow that at 37 degrees because most pathogens are dimorphic. So that is a lot of information. I know it is. But like I said, you could take an entire college course just on mycology. So that this is really a brief touch on this subject. But important and interesting stuff, and you will come across this in the clinical lab. I hope that that all made sense, and I'll see you next time.